up everyone, I'm Eldren and today we're breaking down proteins. Proteins are the most abundant and functionally versatile cellular macromolecules. While every cell in our body has the same DNA in it, it is the proteins that make each of our cell type different from each other. That is why our hair is not the same as our eyes and as our nose. Proteins are long molecules that have a specific three-dimensional shape, and what proteins can do is a lot. Proteins can cause biochemical reactions, act as a messenger, provide structure, transport and store nutrients, provides energy, and many other more. Proteins are necessary for our survival. We have to eat them to be able to create them in our own bodies. Into proteins, structure is everything. From their most basic to complex levels, structure determines the function of proteins. Many of the foods we eat contain proteins, which consist of amino acids. There are hundreds of amino acids found throughout nature, but only instructions for 20 are contained in our genetic code. The amino acids that organisms can biosynthesize differ. Some amino acids must be provided by diet. These are called essential amino acids. Proteins are created by connecting amino acids end-to-end like the links in a chain, with chemical interactions and bonds within the chain cause proteins to fold into knot-like formations. Proteins have four structural levels: the primary chain of amino acids, secondary is the formation of patterns within the protein, tertiary are the side chain interactions leading to the stability of a single protein molecule, and quaternary is the association of multiple protein molecules or subunits. And if they experience conditions outside their normal range, they'll denature or they will fall apart. In order to be functional, protein must maintain its three-dimensional shape. When a protein loses that three-dimensional shape, we say that it has been denatured, and denatured protein is no longer functional. Three-dimensional structures of proteins are held together primarily by hydrogen bonding and London forces. So when we introduce reagents into a protein that interrupts these non-covalent interactions, the protein loses its three-dimensional structure and normally will fall out of solution. You've seen that when you make eggs. There are different methods that can be used to break the bonds. That is why in this experiment, we're going to denature proteins using heat, acid, and alcohol. Now let's get breaking. In breaking proteins, we're going to need a few things. We need 4 glasses, a bowl and spoon, 10 ml syringe, hot water, vinegar, alcohol, and finally, eggs. First, we prepare all the materials we need. We're going to use a bowl for holding our proteins. For looking at them, we're going to use these glasses. These are heat-safe glasses so that we can actually see what is going on as we break our proteins. We will use hot water, vinegar, and ethyl alcohol. We also need our proteins, so today, we'll be using eggs. Alright, let's get started. Today we're going to use just the whites of the eggs because that is where the albumin, the protein we're interested in breaking. So we're going to separate the whites from the yolk. One of the things we want to do is have a control. So we're going to start with one condition where we don't add anything but our egg whites. Our second condition, we're going to add hot water. For our third condition, we're going to add vinegar. And for the last condition, we're going to add ethyl alcohol. One of the things that you might want to do when you're doing this experiment is to label your conditions so that you know exactly what they are. Because right now, all this liquid looks remarkably alike. Now that we have our conditions labeled, next we use the syringe to transfer 5 ml of egg whites into each of our conditions at the same time. So one of the things you can do is to let your proteins sit in a different conditions, or if you would like to see what happens, you can stir them and observe the result. And that right there are denatured proteins. So we have our 4 conditions now, and let's think about what's going on in them. In our control, we've not added anything to our egg whites. and that means the albumin or the proteins are still tightly bound up in their specific three-dimensional structure. In hot water, heat can be used to disrupt hydrogen bonds and nonpolar hydrophobic interactions. This occurs because heat increases the kinetic energy and causes the molecules to vibrate so rapidly and violently that the bonds are disrupted. The proteins in eggs denature and coagulate. In vinegar, the protein becomes denatured when its normal shape gets deformed because some of the hydrogen bonds are broken. Weak hydrogen bonds break when they are exposed to an acid, just like the vinegar. And lastly, alcohol also denatures proteins. It does this the same way as heat, by breaking the bonds that hold parts of the protein in a folded shape. Sometimes the alcohol molecules bond directly to some of the parts of the protein, disrupting the normal way the protein would bond to itself. So alcohol is called a bond disruptor. The proteins again unfolded, taking up more space and hardening into place next to one another. Thank you for breaking things with me today, and I hope you get a chance to break things at home soon.